Hello, thank you for tuning in to Science with Chris. Today I'm going to be discussing Parkinson's disease. Specifically, I'm going to be referring to this Paris ZNF746 repression of PGC1 alpha contributes to neurodegeneration in Parkinson's disease. So for my introduction into this paper, there are roughly 200,000 new cases of Parkinson's disease every year. And Parkinson's disease is a central nervous system disease with symptoms surrounding general muscle difficulties, such as stiffness, tremors in the hands, as well as loss of balance and coordination. For this disease, there are currently no treatments available to cure it, though there are certain antivirals or physical therapy as well that can help combat some of the symptoms of Parkinson's disease.
Parkinson's disease can be formed in two different ways: sporadic and autosomal recessive. 90% of Parkinson's disease cases are sporadic and 10% are autosomal recessive.
Both cases cause loss of dopaminergic neurons in the substantia nigra located in the midbrain.
To start the paper off, they're going off of previous work in this Dawson lab, and Dawson labs at Johns Hopkins. And in this lab, they did like a huge screening, and they're trying to figure out using Parkin as bait. Parkin is the protein, like Parkin, and then Parkinson's disease. So that's how Parkin's related to that. But anyways, so they're using Parkin as bait, and they're like sprinkling Parkin all over the place, and they're seeing which proteins will be able to interact with Parkin. And this was done through a yeast 2 hybrid study in 2000 within the same lab, and they identified a Parkin interacting substrate, Paris. They interacted Paris, and there's like zinc finger 746, and so they have a lead. And their job is to try and connect how Parkin, which is one of several ways that you can get Parkinson's disease, which is the death of dopaminergic neurons in the substantia nigra. There's also alpha-synuclein and other proteins and other gene mutations that will cause Parkinson's disease, but for this paper we're focusing on Parkin. So they have Parkin, and they have this new protein called Paris. And this was in yeast, so they next want to check to see in regular mice, C57 black 6, your good old mice, if there is presence of Paris there. So they checked through Western blot. They're able to see, okay, cool, there's Paris in these brains, and it's uniquely expressed in certain regions, i.e. hippocampus. And then they check, okay, in the yeast, these things bound together. Is this also true within mouse brain? And this is the legit stuff, so they're looking at it, and they're able to see that they co-localize. At the same spot where there's Parkin, there's the exact same spot as Paris. So that gives more interest to this protein that then goes on to explain the rest of the story. So moving on to that, they want to check this again in another cell line that they're going to be using within this paper and for also future references. They use a human neuroblast cell line, and they're able to see that when they have Parkin, Paris is there as well. So this is double confirmation, so the paper is really ready to start figuring out the biological importance of Paris within these neurons. So the first thing they check to see is how does Parkin affect Paris? And Parkin, a little bit of backstory, Parkin is an E3 ubiquitin ligase. Its job is to put these small subunits called ubiquitin onto proteins. And by putting enough ubiquitin subunits onto a protein, it identifies that protein as something that should be degraded by the cell. So you can think of it as like something like targets proteins for destruction. And we wanted to see, well, the Dawson lab wanted to see if Parkin was doing the exact same thing with this Paris protein. And they're able to show that Parkin is doing the same role as it does for other proteins as it is doing with Paris. So that's interesting. So now we know that. And they also do other tests as well, but they're able to show that Parkin is sufficient for controlling and regulating the levels of Paris within the cells. And it's also necessary. So hence Parkin can do the job of regulating the amount of Paris, and it's also necessary. So without Parkin, you're not going to be able to regulate Paris quantities within the cell. So now we have this relationship where Parkin is controlling Paris levels. So they want to see, okay, well, is Paris upregulated, or is there an increased level of Paris within Parkinson's disease brains in mice? They're able to show that with either sporadic cases of Parkinson's disease or autosomal recessive cases, that there is a huge influx in the amount of Paris present in these cells. So hence, because Parkin is not able to control the levels of Paris goes crazy. Like, Parkin is a wolf, Paris is like a rabbit, and if you have no more wolves, then the rabbit population is going to go insane. So that's kind of how it's working out. So super interesting. Paris levels are important, and they're shown that within sporadic PD cases and autosomal recessive that there will be an increased level of Paris. So now they're getting the picture of the puzzle together. So they got the first few steps, but now they're wanting to see, so how does an increase of Paris relate to Parkinson's disease? Like, what's the biological mechanism that goes through those steps? And so they like mold Paris. They have Paris and they mold all around it to figure out all of the like binding areas on this protein. And they figure out a few interesting places. And with these places, they go through like a huge database. And with the database, they're able to show that some of these areas specifically regulate other protein or other gene expressions. So specifically, with high levels of Paris, you're going to have a decreased level of PGC1 alpha. And then you have PGC1 alpha, and that controls a whole slew of other genes. Specifically, one of them was NRF1. And the interesting thing about these two is that they're responsible for maintaining a functional and healthy mitochondria. And you can ask Austin Solomon what the mitochondria does in the cell, and he'll tell you it's the powerhouse of the cell. And he's exactly correct, because the mitochondria makes ATP for the cell using proton gradients and this and that. But without a mitochondria, you're not going to be able to create energy. So you have a decrease in the ability of mitochondria to function, so they start dying off. The cell's not able to have enough energy, so the cell dies and decays, and thus you have how a modification or destruction of the amount of Parkin linking to the destruction of a dopaminergic neuron within a certain part of the brain. So that's what this paper really kind of laid the groundwork for. They really explained the biological mechanism using Paris as this new Parkin interacting substrate.